Hello, good evening. I would just like to know that whether I am audible, and I know the board is not visible. I'm so sorry, but at least it is visible to some extent. Am I audible? I would just like to know whether I am audible. If anyone who has joined could please tell from where you have joined and whether I am audible. Can you please comment that whether I am audible and you can see the board? Maybe you may not be able to read what is written on the board as I can see. I'm so sorry for that, but still, at least can you hear me? Can anyone please comment and tell that whether I'm audible? Yes, please. Yeah, thank you. So we'll wait for a moment if anyone joins, or otherwise we'll go ahead. Yes, thank you, ma'am. Oh, thank you, Komal. Thank you, Reva. Yes, we'll just wait for a moment if anyone joins in, and or otherwise we'll continue. We are not going to waste too much of time here. I think without wasting much time, we should start because it's already too late for me to start the session, right? So yes, we all know that what we had been awaiting for all these days. I don't know whether you were awaiting for that or not, but then what we had been, many people around the globe had been awaiting for, that is your Nobel Prize in physiology and medicine, right? The field where we all are planning to make our careers, right? People are working day and night hard to discover new things in the field of science, right? And it is this particular Nobel Prize, right, which we all really await all these years. I really don't know where I kept my marker. I'm trying to find that, actually. Excuse me for that. I don't know where I kept it. I just wrote it and I don't know where I kept it. Just give me a moment. Just a moment. I don't know. Muskan, can you please see the marker? I don't know. I kept it. I don't know where I kept the marker after I finished writing. Anyway, so this year's Nobel Prize, right, for physiology or medicine, right, it went to your Victor Ambros and what is called as your Gary Ruvkun, right? So Victor Ambros and Gary Ruvkun, right? They are awarded this year's Nobel Prize in 2024 for the discovery of micro RNAs and their role in the post-transcriptional gene regulation, right? A lot of molecular biology out there, a lot of DNA, RNA, and the proteins out there. Anyway, so this year's Nobel Prize for this DNA and RNA and a lot of molecular biology stuff, where they have tried to find out that what exactly was the role of these small RNA molecules in regulating the expression of your genes, right? What intrigued these particular people, the Victor Ambros and Gary Ruvkun, was how all the cells, like which all with we are made of, we are made of so many trillions of cells, and we all are made of same kinds of cells, right? Not all same kinds as such, but then yes, all the cells which are there, they have the same information. Different cells, you have muscle cells, you have nerve cells, you have, I don't know, all sorts of cells, epithelial cells, and all of them are having the same information. All of them are having the same DNA, but then all are having different characters, right? Your muscle cells are contracting, your nerve cells are conducting impulses, your intestinal cells are absorbing, they're digesting. So different cells, they are performing different functions, right? So you have different cells, a variety of cells, but then still the information which in them is there, it is exactly the same, right? And one particular thing which could do that was what is called as your gene regulation, right? So the only thing which could do this was if they could get a proper instruction and the correct set of genes which were there, they would be expressed, right? So we have same genes, right? We have everything out there, but then what to do when and how, right? That is what to be controlled. So this particular phenomenon. So this particular phenomenon I told of gene regulation, it really is the one which actually functions in seeing that the cells, they are really acting different, right? So they are going to act different. They are having the same information, but then yes, which is going to be expressed when it really needs to be taken care of, right? So every cell, the correct set of genes which are there, they are active at a particular time. And it was these two people, Victor Ambros and Gary Ruvkun, who told that is the microRNAs. I'm so sorry, I'm not getting any marker here. It is, but you still focus on what I'm saying, right? The microRNAs, right, which are playing a crucial role. And that gave a new principle of what is called as your gene regulation, right? So it is these, the small RNAs, and we're surprised to know that the human genome, it comprises of thousands of microRNAs. I'm so sorry. So these particular thousands of microRNAs, right? These are, yes, great. So these thousands of your microRNAs, they are actually giving a new dimension to this, the phenomenon of gene regulation, right? So they regulate, they are actually turning it on, they are actually turning it off, and they are trying to decide that how exactly the genes, they are functioning, and how the development it took place, and how the functions of a different cells, they are controlled, right? So essentially, matlab, you find that the genes, they are regulated. We all have learned that DNA is what you call as converted to, not converted to, but the information in the DNA is read into the information of RNA, and it is the information of RNA that is what is called as your translated and sent to your machinery in the cell, in the cytoplasm, to form proteins, right? So the two steps of transcription and translation, where your genes are being expressed, right? So we have different organs which are made of different tissues, and it is these particular tissues which are made of different cells, but then again the cells, they have identical genetic information. But different cells, right, they are performing different function. And what is required is the control of the gene activity, right? You need to control the activity of the genes, and you also need to fine tune these particular activities. So for this, matlab, it is this RNA which is playing a major role. And if anything goes awry in this regulation, kuch gadbad ho gayi is regulation mein, you'll find that leads to disorders like your cancer, it leads to diabetes, it leads to autoimmune disorders, and surely we don't want that. So these are being what you call as done, and it's not something which is happening right now. It was laid back in 1960s, where various transcription factors, they came into picture, and it was these transcription factors, right, which have what you call as which have been known to regulate the expression of a particular gene, which will decide which mRNA will be produced. And then once people came to know that yes, there are certain transcription factors which would decide which mRNA is going to be produced, probably they thought the mystery was solved, because now you could control, like which particular mRNA is being produced, and probably then you can decide the transcriptional control of that particular gene. But then still, it was in 1993, the of article by these two, Victor Ambros and Gary Ruvkun, they completely had a new level of what you call as regulation. So basically what I'm trying to say is ki, you have a DNA, then you have a RNA, and then you have a protein. And now you want to decide which protein is going to be made. So you're going to decide which RNA is going to be made. This was one level of regulation. That is what is called as transcriptional control. Probably you learned at the levels of your operons and all, where your repressors were trying to what you call as stop the synthesis of a particular mRNA, and that all stuff, like, so that is one thing. And Victor Ambros and your Gary Ruvkun have come up with small RNA molecules which are controlling the genes at the next level, right? So you want to fine tune that, right? You want to fine tune it. You want to regulate the activity. You want to turn it on. You want to turn it off, and then you also want to fine tune that. So how exactly this is done, right? These people were trying to do that, and it was this Victor Ambros and Gary Ruvkun who worked with this small worm, C. elegans, right? The Cerebrotis elegans, the model organisms used also in the Human Genome Project, if you remember, right? So this small worm was responsible for this big breakthrough. So this small worm Cerebrotis elegans came up breakthrough late in 1980s when your Victor Ambros and Gary Ruvkun, they were working as postdoctoral fellows at the lab of Robert Hurwitz, who also received a Nobel Prize along with two other people in 2002, I suppose, right? They worked with this 1 millimeter worm. 1 millimeter worm is going to change the entire history. So this 1 millimeter worm itself is only of 1 millimeter, but it is having all sorts of cells. It is having the nerve cells, it is having the muscle cells, and almost everything what is happening inside a particular human, you can say, like it is almost like not everything. So all sorts of cells, they are found in this one particular worm. They were trying to find out the genes which are actually controlling the time of your genetic program, and the expression of the genes at the right time in the right amount, that was the crucial factor in development and growth of these particular worms. These two people, they identified, the Victor Ambros and Gary Ruvkun, two particular mutants, that is LIN4 and LIN14, which had defects in this proper timing, and therefore the defects in their development. And then they identified this mutation. It was also known by them that this LIN4 is a negative, what you call as a regulator of your LIN14. But then how exactly this was working? This they didn't know. They knew that something like somehow LIN4 mutant is actually not mutant actually, but LIN4 is regulating, negatively regulating what you call as your LIN14, right? So control was from LIN4, and what was being controlled was your LIN14. But the question was how? Control to ho raha hai, par kaise? Like how exactly this control is there? Ambros, he found this LIN4 clone gene, which was now producing unusually short RNA. And for his surprise, he found that this LIN4, which was controlling LIN14, when a particular segment of gene is controlling other segment of gene, we generally think that this will produce a protein, and that protein will either stop the what you call as transcription, as a transcription factor. But to his surprise, he found that LIN4 was producing unusual short RNA, which did not code for any protein. So the small size LIN4 was inhibiting LIN14, and it was Gary Ruvkun who later found that LIN14 production of your mRNA from LIN14 was not inhibited by your LIN4. That means LIN4, though it was being produced, it was not inhibiting the production of mRNA. Wait, LIN4 is stopping the expression of LIN14 gene. Okay. LIN4 is not producing a protein. Okay. In presence of LIN4, LIN14 is also not producing a protein. Okay. But in presence of LIN4, LIN14 mRNA is also being produced. Now that was creating a confusion. Now already you are more messed up. So what this particular person found, that it was production of mRNA which was not hindered. So even if in presence of LIN4, you see LIN14 mRNA is being produced, but protein is not being produced. It means that the regulation or inhibition is at the later stage, not at the production of mRNA at least, right? So LIN4 sequence, it matched the complementary sequence of your critical LIN14 mRNA. So you take the sequence of LIN4, you take the LIN14 sequence, and they were complementary to each other. So maybe this LIN4 used to bind this LIN14 and it used to stop its expression. So this is how LIN4 was actually regulating the expression of LIN14, right? A wonderful discovery, a role of micro mRNA came, not mRNA, micro RNA came into picture, and this blocked what is called as your LIN14. So LIN4 is a small micro RNA which is controlling the expression of your LIN14, and this micro mRNA, not mRNA, I'm so sorry, this micro RNA was discovered and it was published first in 1993 by people in the journal Cell. But many people, they didn't pay heed to this. They thought that probably whatever expression of the genes is taking place in C. elegans is not very relevant to humans. They thought that this is a mechanism which would be more limited to your C. elegans. But then you will find that it was in around 2000, in the year 2000, where Gary Ruvkun, he came up and with another particular gene, that is LET7. And this LET7 gene, the micro RNA which was there, it was conserved animal kingdom. So you have all the animals in the animal kingdom having this micro RNA gene, that is your LET7. And then after that, we have identified thousands of your micro RNA genes, thousands of different genes with different micro RNAs. They are now universally conserved and they have a universal mechanism. Many people throughout the globe then started working on how microRNAs are formed, how these microRNAs are delivered at the target site, how they inhibit it, or how they degrade the target RNAs. It is also observed that a single microRNA can regulate multiple, what you call as your expression of many genes. So you have one microRNA controlling the expression of many genes. At the same time, you have what you call as a single gene, which is by the control of many microRNAs. So intriguing mechanism, right? So probably these are helping us to protect plants against inspection. Long back, you remember Andrew Z. Fire and Craig Mello awarded Nobel Prize in 2006 for the phenomenon of this RNA interference. And this tiny RNA, this small RNA, what you call has had a great role in the physiology. So the RNA is small, but the physiology is great. So a tiny RNA with a profound physiology, where hundreds of millions of years this phenomenon is. People say that this particular phenomenon has been conserved by hundreds and millions of years, and it has actually shaped and enabled the evolution. If there is any abnormality in this particular consensus, you will find it may lead to cancer, it may lead to hearing loss, it may lead to eye loss, it may lead to skeletal disorders, it may lead to like many things. So mutations in your microRNA production also results in production of this Dicer-1 syndrome, which is responsible for cancer of various organs and your tissues. So Victor Ambros and Gary Ruvkun has opened new dimensions entirely for the gene. So this is what I wanted to share with you all, like how exactly this Nobel laureates of 2024 in physiology and medicine were awarded this prize for their discovery of microRNAs and the role in post-transcriptional gene regulation. So to just summarize in brief, there are small RNA molecules which are actually controlling the expression of more RNA molecules, and they are actually shaping our evolution and development. And this is really going to have a great impact in the human lives in the coming years. Thank you so much. Good night, and thank you so much for patient listening. And I hope that you will share it with your loved ones who really love. Maybe they are not your loved ones, but they have love for science. Maybe I'm planning to come up with a Hindi version of this video. So if you really think that Hindi version will really help, so please comment here. And thank you so much. Bye bye. Good night. Thanks. I really hope only very few people who joined, they really came to know something about this. I hope the session helped you all to know about the recent discoveries of Gary Ruvkun and Victor Ambros, the Nobel laureates of 2024. I hope this helped. Yes or no? If you could please comment, that would really help me to make more such videos. Thank you. Good night.